Greetings. My name is Elizabeth Gwartney, and today I'll be presenting my research on novel antibiotics. Greetings. My name is Elizabeth Gwartney, and today I will be presenting on my research over novel antibiotics that I have found in samples from Oklahoma soil. Antibiotic resistance is a major crisis in the United States. According to the Centers for Disease Control and Prevention, the CDC, in 2018, more than 2.8 million antibiotic-resistant infections had occurred in the United States each year on average. As a result of these infections, more than 35,000 people have died. Among the most well-known of these infectious agents is methicillin-resistant Staphylococcus aureus, or MRSA, which killed 20,000 people in the United States in 2019 as a result of bloodstream infections. We have also seen a 4% increase in non-healthcare associated infections of MRSA, which indicates that this pathogen is now spreading in the community. This graphic shows a visual representation of common antibiotics and the microorganisms that they cover. As you can see, penicillin at the top covers most gram-positive organisms, while amoxicillin and ampicillin cover some gram-positives and some gram-negatives. Vancomycin and daptomycin are common antibiotics used to cover methicillin-resistant Staphylococcus aureus, but as you can see, they do not have coverage against Pseudomonas or anaerobic organisms, which are also common infections. This indicates that it is very important to choose antibiotics wisely. For the purposes of our research, we defined an antibiotic as a substance secreted by a microorganism that inhibits the growth of or kills another microorganism. This is a common feature found in soil microbes because they have to compete with other microorganisms for resources in the ecosystem. The most common example of this is Penicillium rubens, which was the first microorganism discovered that produced an antibiotic, penicillin. The solutions to the antibiotic resistance crisis are first and foremost improved hygiene practices, such as hand washing. Infection prevention in healthcare settings is also very important, as healthcare infections are more likely to become resistant to antibiotics. Good antibiotic stewardship is also important so that we don't prematurely develop resistance in certain species. The last resort solution to this problem is the identification of new antibiotics, which is the focus of my research. Our experimental approach was to first collect soil samples from this garden. As you can see, it is very green, lush, and healthy, which indicates good soil quality. We diluted the samples with sterile saline and plated on media designed to allow the growth of different types of microorganisms, such as bacteria and fungi. We then incubated these at 27 degrees Celsius, which is different from the typical 37 degrees Celsius of normal human body temperature. We chose 27 degrees because it is closer to the conditions that these microbes would naturally grow at. We then selected individual colonies that looked interesting, had interesting morphology, and screened them for antibiotic production. This is an example of the assay that we designed to detect antibiotic production. It is a variation on the Kirby-Bauer assay, which is designed to test sensitivity of certain microorganisms to antibiotics. First, we spread the plate with a pathogen of interest. In this example, we used E. coli. We removed a core from the middle of the pathogen plate and cored out a sample of the garden microbe from the plate that it was growing on. We transplanted the garden microbe into the pathogen and sealed it with top agar, which is just a liquefied version of the growth medium. We then incubated these at 27 degrees Celsius. As you can see in this example, the microorganism in the middle is secreting a substance that is inhibiting the growth of the E. coli around it. This image shows the 4 microorganisms that we identified that showed the most promise as antibiotic producers. We tested these against Shigella, which is also a common pathogen that develops microorganism antibiotic resistance. CRB1, CRB2, CRB3, and CRB4 are the working names of these microorganisms that we have discovered. As you can see, CRB1 and CRB4 show no inhibition of the growth of Shigella. However, CRB3 shows very good inhibition of the growth of the Shigella. CRB2 shows mild inhibition and is worth studying further in this microorganism. This slide shows quantification of our inhibition of Staphylococcus aureus in 3 of the species. CRB4 did not show any inhibition activity against Staphylococcus aureus. The bar on the far right indicates our control, which is the diameter of the core itself, 13 millimeters. We have tested these organisms against Staphylococcus aureus in multiple assays, and the bars show the average inhibition against Staphylococcus aureus, while the error bars show our standard deviation in measurements. According to a 2019 CDC report on microorganism antibiotic resistance, these are classified as urgent threats to the healthcare system. These are also microorganisms which we have shown great effectiveness against. Salmonella typhi and Shigella are microorganisms that we can work with in the lab, as well as Streptococcus species, although we cannot work with pathogenic substances. We do not have access to methicillin-resistant species of Staphylococcus aureus. However, we have access to many Staphylococcus aureus species, and we have shown effectiveness against all of them. We are also not allowed to work with Mycobacterium tuberculosis. However, we can work with different Mycobacterium species and have shown effectiveness against those. In order to learn more about these microorganisms, we conducted some diagnostic stains. This is a picture of an endospore stain. The green dots in the middle of the red bacteria indicate the presence of spores. Spores are common in bacteria that are found in soil because they are very resistant to differences in condition. The most common example of a sporulating bacteria is Bacillus anthracis, which is the causative agent of anthrax. CRB1 and CRB2 both have a positive endospore stain. This particular image is of CRB1, and you can see the little green dots in the middle of the bacteria, which are pink, indicate that they are producing spores. This is an image of a gram stain of CRB3. A gram stain is a common diagnostic stain used in medicine to determine the cell wall characteristics of bacteria, and it is often one of the first stains performed to decide which antibiotic to choose to treat which infections. CRB1, CRB2, CRB3, and CRB4 are all gram-positive. This backs up our endospore findings in CRB1 and CRB2 because in order to be sporulating, a bacteria must be gram-positive. Next, we decided to sequence the 16S ribosomal RNA gene in all of our microorganisms. This gene is highly conserved among prokaryotes and allows us to identify what these microbes might be related to or their exact species. These specimens were sequenced by GeneWiz. The resulting sequences were then input into Basic Local Alignment Search Tool, or BLAST software, from the National Institutes of Health, and this was also used to compare sequences. This is a partial histogram of the 16S ribosomal RNA sequence of CRB1. As you can see, each peak represents the presence of a certain nucleotide. The letters on the second row from the top indicate which nucleotide was present in each peak. The letters above that indicate which amino acid this sequence codes for. This is a partial histogram of the 16S ribosomal RNA of CRB3. As you can see, the results are not as clear-cut. This indicates that this organism is likely a eukaryote and not a prokaryote because there was not a 16S ribosomal RNA sequence for the machine to read. The ends in the genome represent an indeterminate sequence identification. Our results indicate that CRB1 is likely a strain of Bacillus subtilis, CRB2 is likely a strain of Bacillus valensins or Bacillus subtilis, and this makes sense considering that Bacillus strains are often sporulating. The results for CRB3 were inconclusive, indicating that it is likely a eukaryotic organism, such as a fungus. CRB4 was identified as a Streptomyces species, although none of these tests indicated a conclusive result as to the exact strain of these microorganisms. For the future, we would like to conduct partial purification of the antibiotics to be able to test their effectiveness further. We would also like to conduct differential media tests on our microorganisms to further learn about their characteristics. We would also like to conduct a targeted sequencing of the genes in order to learn more about the type of organisms that we have found. So far, we have identified 4 microorganisms that secrete antibiotics. 3 are related to known bacteria, and the 4th is likely a fungus. I would like to acknowledge Greg Mullen, my research advisor, and Beth Landon, our program technician, for their assistance in my research. I would also like to thank the Beta 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 Research Foundation for their grant support of my research. Thank you.